Um, I can't see you all, but hopefully I'll see some of your faces when it comes to questions. We were just discussing, if there's something that you want to know during the presentation, feel free to interrupt, and I'll do my best to answer on the spot. All right, so let me share my presentation. Okay, does this look all right?
It's great.
Yeah. Okay, awesome. All right, so today I'd like to talk to you about some of our recent work on quantum biosensing, and in particular how we can try and look at multiple different modalities at the same time using NV centers in diamond nanocrystals. And so as a little motivation here, some of the questions that we're interested in are around temperature in living systems, particularly at the nanometer scale. So if you think about it, measuring temperature is actually quite difficult if you think of sort of prodding in a thermometer into something that's nanometer sized. Obviously, you're bringing a big thermal load into the system, and so measuring at that length scale is extremely difficult. And there are a lot of interesting questions around that topic, even on the fact of what the definition of temperature is at that length scale, because it's a thermodynamic property which relies on large ensembles. We're looking at very small volumes here. And then also in living systems in particular, what kinds of temperature gradients are there? Are there any sustained temperature gradients inside a cell? Is the mitochondrion, for example, hotter than the rest of it as it's producing all of this energy? So there's a lot of sort of metabolic dynamics there that we just don't understand because we don't have access to thermometers at that length scale. Another question which is related to that is, how does temperature affect viscosity? So we know that there's a phenomenon called viscoadaptation in cells, where when the temperature changes, of course, that would speed up chemical reactions. Most of the motion in cells, chemicals reacting with each other, still happens through diffusion. There are a lot of active processes, but a lot of it happens through diffusion. And if you raise the temperature, then of course the reactions will speed up, and this might not be something that the organism wants. So there are ways for it to regulate the viscoelasticity of its interior. But again, very little is understood on this because it is so hard to measure on those length scales. And a final example, looking at slightly larger systems, how does an entire organism respond to stress? So, for example, if we take very basic animals such as C. elegans worms, we perturb them through genetic or chemical thermal perturbations, what happens to the viscosity? What happens to the development of this animal? Can we understand something about how the geometry of an animal is formed and how this can be regulated and how robust it is to perturbations, which of course is an interesting question relating to sort of climate change and changing environments. How will animals adapt in the sort of early embryogenesis process to these changes? So these are some of the questions that we're interested in this space. And of course, ideally, we would like a nanoscale sensor that can measure temperature at the millikelvin scale. That would be amazing. And then we would also like it to be able to perform rheology, so measuring viscoelasticity at the nanometer scale. And this relates to many diseases, of course, as well. So as well as being interesting from a sort of fundamental biology perspective, many diseases have changes in viscoelasticity inside the cells. So, for example, we know that cancerous cells become stiffer as the cancer progresses, and there are many links to other diseases as well, including viral replication, for example, in sort of denser liquid droplets. That would be excellent to understand so that we could find better treatments for these diseases. And as I mentioned, measuring multiple parameters at the same time to really understand early development of slightly more complex organisms. All right, so what do we need? We need a sensor that's minimally invasive, can operate at the nanometer scale, and ideally can probe multiple parameters at the same time to reveal these complex interdependencies that occur in biological systems. Of course, our solution is the NV center in diamond, otherwise I wouldn't be here talking to you today. And I'm sure you're aware of its very promising properties, which include low toxicity. So diamond itself is very inert, so it doesn't tend to perturb biological systems much, which is very nice for a sensor. You want to be minimally invasive. It is amenable to surface functionalization, though, if you get the chemistry right. And of course, being so small, it can be embedded in 3D structures, which is very nice for intracellular sensing. So I want to start by thanking the team behind this effort. So we've sort of got a number of areas in the group, but about half of the team is working on the sort of biosensing side. You can see here on the right side. Let me just get the laser pointer. There we go. So this is our bio team here. So Gu, who was a PhD student, is now at MIT. And we have Jack, who's the leading postdoc on this project, and 3 PhD students. This is also a collaboration with Mette Atatürk in physics, Liliana Fruck in chemical engineering, and Eric Mishka on the C. elegans worm side at the Gurdon Institute here in Cambridge. So I'll be showing work from Jack, Louise, Sophia, Julien, and Qiushi today. I wanted to quickly highlight some of the other things that are going on as well, in case this is of interest. Send me an email. I'm happy to talk more about this. So we also look at NMR and ESR, so nuclear magnetic resonance and electron spin resonance using spins in diamond close to surfaces. And we have a project that looks at complex interacting spin systems, where we're building a quantum simulator based on spins in diamond. So quite different topics, but today we're going to focus on the sensing in living systems. So here's an outline of what I want to talk about today. So I'll start with introducing the sensor and its modalities. Even though this is probably familiar to a lot of you, hopefully there'll be a little bit of new information there as well. And then I will introduce our quantum biosensing chip, our cubic chip. This is something we've been developing over the years, and we're about to publish a paper that really contains all of the details. So this is sort of a sneak peek into some of those properties. And then I want to follow on with some sensing experiments we've done in living cells, simultaneous thermometry and rheometry in living cells. And finally, I want to highlight a couple of opportunities, in particular on the C. elegans side, looking at slightly larger organisms and what we are hoping to learn there. All right, so the nitrogen-vacancy center, I'm sure you all know, substitutional nitrogen next to a vacant site, which conveniently has a spin structure that we can address through optics and microwaves. We typically use nanodiamonds that are sort of between maybe 50 and 100 or so nanometers in size and contain either a single NV center or an ensemble of NV centers going up to sort of hundreds of NV centers per nanodiamond. In terms of the level structure, here you've got the diamond bandgap, which is conveniently very wide, and it's a very nice host material for this defect. And you can see here the sort of our qubit manifold, the spin triplet ground state here, which is split by 2.9 gigahertz microwave transition. And of course, the exceptional thing about the NV center is that we can initialize the spin using optics, using green excitation typically. And you can see as if you follow the arrows around, whichever spin state you start out in, you'll always end up in with higher probability in the MS equals 0 state here. And so that's how spin initialization works. We then have the radiative decay here, which only originates or predominantly originates from the MS0 state and tells us whether we were initially in the 0 state or the plus and minus 1 state. So we call this 0 state the bright state, which is why it's got a yellow glow on it here in the diagram. So if we get many photons out, we know that we were in the MS0 state. And if we get fewer photons out, we know that we started in the plus and minus 1 state. So that's our spin readout. And then, of course, applying a microwave transition here, we can transfer the spin population from the 0 state to the 1 state and back. And this allows full control of this spin manifold. So if we do a sort of typical experiment called optically detected magnetic resonance, we can sweep this microwave frequency, as you see here on the right. We can sweep the frequency, and if we're monitoring the number of photons that we collect from this defect, we'll see a drop as we hit the right frequency to transfer population from the bright state to the dark state. Now, why is this interesting? Well, it turns out that actually a lot of the quantum sensing schemes, especially in biological systems, are based on exactly this technique. And so if we apply a perturbation, in this case, it's an external magnetic field, the plus and minus 1 levels split apart, and we will see this as a feature in the resonance. And of course, from this energy level, from the splitting of these resonances, we can extract the energy level spacing, which then relates to the magnitude of the perturbation. So this is an example for magnetic field, but it turns out temperature through the expansion and contraction of the diamond lattice also gives us a change in these energy levels. So they shift in a slightly different way. They don't shift apart, they shift up and down, which allows us to distinguish those two external perturbations, which is kind of the basis for some of the multimodal sensing aspects. So we can really distinguish which external parameter changed in a given instance. So quick summary, we've got a spin triplet ground state. We can control it through optics and microwave excitation. Through these nice decay pathways, we have optical initialization and optical readout of the spin state. And it's a rather unusual quantum system in that it's solid state based and it works at room temperature, which means that you can put it into messy systems such as biological and living wet systems. Of course, that was the most sort of simple scheme that you can use to do sensing. We can do more complicated things. We can look at pulsed operations, so we can create superposition states. For example, in this experiment, we can apply a pi-half pulse, generate a superposition state, and watch it evolve in time under the effect of, say, an external magnetic field or maybe a change in temperature. So these pulse techniques can typically give better sensitivity. They're a little bit trickier to realize and have some pre-requirements, but there are some benefits to using these as well. So next I want to go through a couple of different modalities, and there are a few, and I'm sure there will be more to come as well. So first of all, temperature, which I already mentioned. As you can see here, this is the sort of simplest way of measuring temperature. You apply continuous microwave excitation, continuous laser to initialize and read out the NV spin. And you see this double dip resonance here, which shifts around as the temperature changes from hot to cold. And we can very nicely step the temperature up and track it. You can see the yellow is a sort of macroscopic large temperature sensor on chip, and the blue dots are what the NV is sensing here. Then what we can also look at is doing this sort of nanoscale ESR that I mentioned briefly in the beginning. So the idea here is that we apply not a continuous excitation, but a so-called spin echo sequence here on the NV center, where we create a superposition state, and then we exchange populations halfway through, and then read out at the end. And now the trick is, if we do simultaneous to this central pi pulse, if we also target another spin species which is nearby, we will selectively introduce noise from only those spins into the spin echo signal. Now, why is this interesting? Well, it allows us, as we sweep this radio wave frequency for the sort of dark spins, the unknown spins, when we hit a resonance, we can see this spin echo of signal of the NV drop, and we can relate the magnitude and the width of that to the concentration, for example, of nearby electronic spins. So this is some rather old data now in a diamond nanocrystal, looking at intrinsic spins inside the diamond. But of course, it would be very exciting to use this to look at spins in a liquid environment, for example, to do spin selective detection of dark spins nearby. Another option, which is sort of taking this a little bit, this technique even further, is using the NV center to look at nuclear spins. So here what we do is we apply a slightly longer pulse train, one of these dynamical decoupling sequences. Here the example I have is an XY8, which is very commonly used for nanoscale NMR. And all you need to do is match the periodicity of these pi pulses with the Lamour frequency of the nuclear spins. And if you do that, again, then you're introducing the noise selectively from that particular nuclear spin species, and you can see a decoherence in the NV center readout. And so you can distinguish between different nuclear spin species. This is, again, data in a nanodiamond. This has been done extremely well as in bulk diamond as well, with shallow NVs. This is a very promising technique, and that there might be some applications for using this in liquid environments in nanodiamonds as well, although there are many technical challenges along the way as well. So that was 3, and there are many more, but I won't go through them in too much detail, except for passive microreology. We will see again, we'll come across again later. NV centers can be used to detect external magnetic fields or voltages by looking at how the charge state changes. So from NV minus to NV plus, NV 0. Of course, we can sense external magnetic fields, which also has interesting biological applications. And looking at the spin relaxation time enables detection of nearby ions and reactive oxygen species, which also has many applications in living systems. All right, so what do? We take this diamond nanocrystal and we introduce it to a living system, and we pick our favorite modality, and off we go. And so if you would like to learn a little bit more about some of these opportunities and modalities, please take a look at our recent reviews that I've highlighted here. I've got one last slide here on this section on some of the things that we're thinking about implementing in the future. So this is very recent data on a diamond nanocrystal looking at a so-called thermal echo, which was introduced in 2013 by Toily et al. And this is a very interesting technique. It relies on a slightly more complex train of pulses than the sort of simple ODMR, optically detected magnetic resonance, but it does allow for much better temperature resolution. So if we're really trying to resolve, do mitochondria actually raise the temperature in their vicinity inside cells, this could be a very promising way to go. And another thing that we're interested in is trying to apply this ESR-based radical detection because it's very robust, because it's so specific. It's a sort of double spin resonance excitation. So we know that we're really only we're getting signal only from the spin that we're targeting. And if there are different spin species, we should be able to distinguish between them. So these are some of the directions that we're interested in. Of course, there are some challenges here. So particularly for the for both actually, coherence times are crucial. So we need high quality diamond, we need low impurities for longer coherence times is very important. And then in particular for the ESR-based radical detection, an external magnetic field that is able to dynamically track the NV quantization axis is also very important, which, depending on the environment that the nanodiamond is in, can be either extremely fast or slow enough that we can actually have an external magnetic field that follows it around. And a couple of things to just highlight. So I'm going to talk about nanodiamonds here for these sort of intracellular and intra-organism sensing applications. But there are a lot of other exciting things that people are doing with diamond in the space of kind of nanodiamonds for biotechnology, healthcare. So I just want to highlight from the McKendry group at UCL, these nanodiamond-based lateral flow tests, which was a really nice result from a couple of years ago, where they got orders of magnitude better sensitivity compared with traditional gold particle-based methods. And then, of course, you can think of sticking a piece of diamond at the end of a sort of laparoscopic probe, for example, and using this to look at magnetic particles in a patient. So some of these applications are really quite close to being applied in a clinical setting, which is quite fun. All right, so next, I want to move on to introducing our cubic chip to you. So this was really developed because, well, we needed a nice chip to be able to do these experiments and not have to kind of rewire everything every single time we do an experiment, to sort of reduce the overhead of doing many repetitions, which of course you want to have for biological systems. You want a lot of data to be able to make statistically significant statements. And so this is what it looks like. So it's a PCB chip, which has this waveguide structure. So we have the microwave lines here. So the coplanar waveguide in green, which delivers the microwaves to the NV centers. And then we have a global heater and a thermometer as well, because we want to be able to step the temperature, control the temperature on a fast sort of minute time scale in a very reproducible way, and measure that on a global scale and compare that with what the nanodiamond is telling us is happening at the nanometer scale. And if any of you have ever tried to do this kind of temperature stepping with the incubator itself, you will know that it's very challenging and it takes forever, and it's not very fun. So that's why we wanted to really have that on chip for better flexibility. And then the other sort of component, which doesn't actually belong to the chip, but which is crucial to some of the experiments we do, is this orbital tracking. And this is because, of course, diamonds, unless they've been functionalized and they're really stuck to something in a cell, they will move around quite a lot. And so during the course of an experiment, you need to be able to follow the diamond around. So this orbital tracking is based on a circular path of the beam that goes around the nanodiamond, which gives us, of course, if it moves away from the diamond, we see the signal drop. And from these kinds of sinusoidal curves and looking at the phase of that curve, we know which direction the nanodiamond moved, and we can make slight modifications to its position, and we can track the nanodiamonds in these systems. And this is also relevant even if you have functionalized the nanodiamond, your cell might be moving around or growing. So even in those cases, this kind of tracking is extremely useful. So what we did was we quantified how sort of homogeneous the magnetic field is. Obviously, we have this active area, and maybe we have a cell culture in that area, and we would like to be able to make full use of it and look at nanodiamonds anywhere in that region. So we want to make sure that we have nice microwave control that's relatively uniform across here. So you can see these crosscuts in both directions in our active area. And this is just an example. This is not the sort of maximal Rabi rates that we can apply. We can go to pretty much whatever is limiting us from the electronic side. And then, of course, calibrating the heaters is another key component. Here what we do is we switch the heater on, so it's just a DC voltage applied to these heating elements, and then we use our resistive thermometer to measure what happens over time. And you can see at lower voltages, this follows very well. The simulations are in the curves, and the data points are measured from the thermometer. At higher temperatures, there are some slightly odd things going on with convection and so on, which are quite hard to simulate. But we can at least measure with our thermometer, and we know what's going on the global scale, on the chip scale. And this allows us to really do this kind of temperature stepping that you can see here. And we get sort of tens of millikelvin RMS stability for our chip, which is quite nice. And it can be changed on the minute timescale if we want to do any kind of stepping. So another thing of concern, and this is something that I often get asked by people who work in this field, but also especially by biologists who say, well, you are applying microwaves to living systems. It sounds a little bit like a microwave oven. What happens? Are they, are they still happy, basically? Are they still alive? Can you claim that this is non-invasive? And so what we did here to check that we were indeed not perturbing the system was look at how healthy these cells are while applying continuous microwave excitation. So here you've got HeLa cells with mitotracker green for labeling the mitochondria, and you can see the nuke blue staining the nucleus. So they're nicely visible there. And then we looked at the mean square displacement of sort of intracellular motions, in particular looking at vesicles inside cells that have a typical MSD when they're alive and happily functioning, and when they start getting a little bit unhappy, it immediately drops. So that's quite a good signature for how happy and healthy the cells are. And you can see here we've got traces, so this is over time with no microwaves, which is our control. And then we start increasing the power, so 14 dBm, 21, etc. And then at some point we get to beyond the threshold where you can see that this MSD plummets and the cell dies. And we can also quantify what kind of a temperature rise that microwave power leads to. So if you look here on the right, we've got the colored lines here, which correspond to the powers we had here. And you can see the temperature rise in degrees that we get from these continuous microwave excitation. So luckily the ODMR experiments that we perform are in this regime. They're sort of below 20 dBm. So we know that we're okay. And of course, for any kind of pulsed operation, the overall power delivered is even lower. But even for the continuous wave experiments, we're in a good regime where don't seem to be perturbing these cells. So that was an important thing to check. All right, so then we've got this chip and it doesn't kill the cells, which is good. So we can move on to sense temperature and viscosity in living cells, which is the next section I want to move on to. So here's an example of a track, a dynamic track inside a cell, where you can see, of course, here the challenges of really keeping track of this diamond nanocrystal because it's moving extremely fast. You can see there are sort of parts that are quite straight, where the diamond is shuttled from one part of the cell to another through active transport. And then there are sort of bits of Brownian transport where it's sort of it's just maybe stuck in a particular area and moving around, just being kicked around by nearby molecules. So there's a lot of interesting dynamics going on here. And one key thing when doing this sensing is to kind of know where you are, because if you only see your diamond nanocrystal and you measure something interesting, if you can't tell where this was measured, then there's very little value in that actual measurement. So actually localizing the diamond nanocrystal to a particular region of a cell is really important. And so what we can do here is a sort of three-dimensional confocal image of a cell. This is a HeLa cell that I'm showing here as an example, where we can see in green the mitochondrial network and the little red dots, which are nanodiamonds that are tucked away and hidden in this mitochondrial network. So we can actually see where these nanodiamonds have ended up. So that's a good start. And then we wanted to really probe that link between temperature and viscosity and see, can we measure those two properties simultaneously in a living system with all its applications then to disease and small organisms. And so we started in a model system, so in glycerol, which has sort of trivial temperature dependence of the viscosity. So you can see here we step the temperature with our chip, and we measure at the nanometer scale what the NV is reporting on temperature, and we can also look at its mean square displacement. And indeed, it starts moving faster as the temperature increases, and this is exactly what we would expect for the medium of glycerol. We can then move to a slightly more complex, still model system. So this is glycerol cross-linked xanthan, which has more of a sort of elastic, stretchy structure and more of an interesting temperature dependence. So you can see here, we can use this mean square displacement data to extract the viscoelastic moduli, G prime and G double prime. And basically what they tell you is how viscous and how elastic the system is. So basically how liquid-like and how solid-like. And you can see the blue stripes here and red correspond to cold and hot. So we step the temperature of the chip between cold and hot. And we see indeed that these viscoelastic moduli step up and down. And so we see changes in the viscoelastic properties, as we would expect. So this works very well in model systems. Then when we move it to cells, this is again looking at HeLa cells. We can do the same kind of temperature stepping. So again, you see the gray line is our global sensor. The red dots are our local NV probe. We can step the temperature up and down, and then we can monitor the mean square displacement or the frequency inverse the power spectral density as a function of time. And the interesting thing here is that we can work out what the sort of thermal baseline is of thermal stochastic forces. So this would be sort of purely Brownian motion. And then we see that, for example, here after about 10 minutes, there seems to be an active transport, an external force that pulls on our diamond nanocrystal. And we can extract the occurrence of these external forces just by looking at this power spectral density here. And by doing the same analysis as we did before, we can again extract the viscoelastic moduli, and we can see that the cell on the nanometer scale locally behaves like a weak gel. So it's more solid-like than liquid-like. It's elasticity dominated in this case. And indeed, that's been confirmed by measurements through other means as well. So this works as a tool set, as a multimodal tool set, and we now want to move on to look at how these kinds of how we can reveal viscoadaptation in an intracellular space. One more slight detail on what else we can extract from this motion. So this is the same track that I showed earlier, and if we take lots of tracks like this and we split them into components that have the directed transport and that have the Brownian transport, we get an interesting distribution here. So this is looking at the mean square displacement. Trying to move the with the log gradient of the mean square displacement, and we can plot that as a histogram, as you can see on the right here. And of course, there's a big difference between the directed and the non-directed parts of the path. And then we can apply nocodazole, which is a chemical that suppresses microtubule polymerization, which entirely stops the directed transport, and it also slows everything else down in the cell as well. And we can look at what these values mean, and indeed this matches really well with the directed transport being super diffusive, the non-directed being pretty much just Brownian. And then when we kind of shut things down, we're in the subdiffusive regime. So again, this is looking at lots of nanodiamonds in the entire cell, but looking at different regions, we can really extract how, what the viscoelastic properties are and what kind of diffusive regime we are in different parts of the cell. Right, so we come to the final part of this presentation, where I want to show a little bit of our recent work on looking at C. elegans worms. So here, of course, the challenge of having a nanodiamond whizzing around a living cell is kind of amplified by having a nanodiamond in a worm that can actually move. So of course, these worms typically sort of swim around, and they're quite hard to catch. You need to sort of really chase them, and so imagining trying to track them on a chip is extremely hard. So what we need to do is we need to somehow immobilize them. So there are chemical ways of immobilizing these worms, but we've decided to go for a mechanical way, so that we are as little invasive as we possibly can, because we don't know what other effects the chemical immobilization might have on the metabolism of this worm. And so my student Sophia came up with this plan to have the worm basically in a sort of ball pit of microbeads. So there's the worm on our cubic chip, and then an agarose pad on top, and it gets immobilized by these microbeads. And this is what it looks like under an optical microscope, and we can indeed we can resolve it in our confocal microscope, and through nano injection techniques and a very skilled hand, you can introduce diamond nanocrystals inside a worm. And so we're able to do the same kind of thermometry in a living worm. And then if you get it into the right part of the worm at the right stage of the worm cycle, you can also introduce these diamond nanocrystals into the C. elegans embryos. And now this is really interesting from the sort of developmental perspective that I mentioned right at the beginning of this talk, where we can look at how viscoelastic changes affect how the organism actually develops. So we know that when the embryo divides, certain parts have different viscoelastic properties, and they develop into different parts of the body of the worm later on. But we don't know how the worm manages to regulate that, and how it is so robust to changes in external temperature, for example. So that's one of the things we're interested in probing. And now, of course, we did our stress testing for cells, but of course we also want to know that these microwaves that we're applying don't perturb the worms. So what we did here, and this is in collaboration with Eric Mishka's group, who have this nice genetic variant of the C. elegans, which expresses fluorescent proteins in different parts of the body. So you can see here the head glows red. This is sort of our control. So every worm will have this red glowing head. And then if the worm gets stressed, it starts expressing green fluorescent protein. So you can see the top worm is very happy, and the bottom worm is not very happy. It's turned a little bit green. So we call these our Hulk worms, because when they get unhappy, they turn green. And so this red part here allows us to sort of normalize the fluorescence from each worm, and then we can control it to the ones that we can use that as a control to normalize the counts in each case. So what you can see here is the fluorescence intensity. So we've got a control, which of course doesn't have any green. And then we apply different microwave powers for 20 minutes duration, and make sure that there is no green, or this, as low levels of green as the control. And we can then also check what happens under a temperature shock, where we know that C. elegans worms are not very happy if you heat them up by, well, above around 35 degrees Celsius. And indeed, we see here that at 33 degrees, there's no green, but as soon as we go higher in temperature, you suddenly see this green cropping up. But the good news is that the microwave powers that we typically use for IODMR do not trigger the stress response. So again, we're good to go with this chip, and we can move ahead with our experiments. So some opportunities in this space is looking at the regulation that these worms use to deal with temperature, viscosity changes, pH changes in the environment. And this could allow us to discover early detection markers for diseases, so perhaps not in worms, but looking at cells. If we can detect multiple parameters at the same time, we might be able to say what an early marker for the onset of cancer is, for example, in the viscoelastic properties at the nanometer scale. And more generally, of course, diamond nanocrystals are well, we know they're great sensors, but they're also they can be used for drug delivery. So this could be quite a nice opportunity to have an integrated drug sensor or integrated drug delivery with a sensor of what the actual response is of an organism to the drug. And of course, there are many other opportunities here looking at other spin-based platforms, which are not diamond, that are sort of coming up. Different materials such as HBN. All right, so that's it from me in terms of content. I just want to leave you with a couple of messages. So nanodiamonds offer robust sensing. They don't bleach over time, which is very nice for looking at sort of whole life cycle measurements of organisms and cells. We can do simultaneous measurements of different parameters, and I showed you examples of temperature and rheology here today. And this could help us uncover interdependencies between different parameters, which are really key for understanding some of the regulatory principles in living systems. And finally, our cubic chip delivers microwaves in a way that doesn't perturb cells or organisms, and is a very versatile tool for these kinds of experiments. And with that, I want to thank you all for your attention, and happy to take any questions.
Helena, thank you very much for a great presentation. It's really very advanced science and underlying technology that you presented today. So we are open to questions. You can either Peter has his question. Yeah, so Peter, please ask your question.
Hi, Helena. My name's Peter Pozowski from the University of Washington. Thank you for the wonderful talk. I was clapping instead of raising my hand through Zoom.
Oh, and now you got pulled up for a question.
But I was really impressed with your results. And I was curious, in terms of the temperature resolution that you're seeing, when you turn on the green pump laser to excite the luminescence from your diamond crystals, I'm curious, can you resolve a temperature change when you turn on your green excitation laser? I'm assuming it's a CW, maybe 532 laser. And I was just curious, is there a potential measurable change in temperature when you excite the nanodiamonds?
Yeah, that's a great question. So I talked about how microwaves can affect the organisms, but I didn't talk about the green laser. And we do see an effect under prolonged excitation, and it is power dependent. So it's not necessarily a rise in temperature. It's more that we can sort of see probably the proteins need denaturing, or there's some kind of process that changes the viscoelastic environment of the cell if we're not careful. So yeah, we need to use it sparingly and at low amplitudes. Yeah, that's absolutely
I was really
right.
impressed, and it's really beautiful data and great work. If no one else has a question at the moment, I had one other question,
Please
which I want.
continue. How many questions you have?
Well, the other question I had was there's some really beautiful work from Taras Plachotnik, I think, and he was in Australia at the time, using the Debye-Waller factor to measure temperature in nanodiamonds, sort of looking at the zero phonon line and compared to the phonon overtones. And I was curious if you may have looked at using the Debye-Waller factor approach just from the luminescent spectrum without microwaves, if your group may have tried that. But I think the ODMR sensitivities you presented are probably much greater or much higher than the Debye-Waller factor.
Yeah, that's another great point. So absolutely, that's another very good way of measuring temperature. And in fact, the sensitivities, I think I can't remember the numbers off the top of my head, but I think compared to CW ODMR, I think they're not that much worse. So it's it's a comparable technique. Obviously, you can't do any of the pulsed thermal echo stuff, so you can't really push it further. The one reason, and if you want to avoid microwaves, obviously that's a really good route to go. We quite like these microwave-based techniques because they offer this added robustness, right? So anything that's purely optical, you run into some of the issues that exist with dyes. So obviously dyes bleach over time. That's one problem you wouldn't have with nanodiamonds. But also, any changes that you're trying to look at, you're trying to just look at the optical spectrum, there are a lot of things that might change that. So for example, changes in refractive index, might change what the spectrum looks like, depending on what kind of molecule drifts in and out of the focal spot. These are very messy dynamical systems. So I think depending on your system, if it's a very clean system, that could work really well. But I think the messier you go and the more kind of other junk you have floating around, the harder it gets to get good sensitivities. Whereas with the microwave control, of course, the only thing that modulates when we change our microwaves on and off is the NV center. And so the refractive index or pH can change all it wants. Our sensitivities won't be affected by that.
Cool. Yeah, thanks again for the great presentation. I really am impressed.
Thank you. Thanks for your questions, Peter.
Any other questions? I will ask a couple questions while people think on their questions. So, Helena, does fluorescent background from biological systems, which can be very high, does it impact any of your results and make more difficult observation? Do you have to do this background-free imaging processing to detect nanodiamonds in parallel to measurements of some physical parameters? So how you take into account that parameter of your biological system?
Yeah, so that's another great question. So we don't actually do any kind of active background suppression techniques. But what we do is we use diamond nanocrystals with many NVs in them. So we have tried with single NV nanodiamonds, which are very nice because you just think about one NV center and, if you want to align a magnetic field, it's very simple. Well, simple in that there's only one direction, and some of the pulse protocols are easier to implement and give better sensitivity. But of course, the counts suffer. And in particular, when trying to track the nanodiamond, if there are a lot of other fluorescent objects nearby, the tracker wouldn't work. So we're really relying on high brightness diamond nanocrystals to make this work.
Mm-hmm. And say in this regard, how critical is size? Say the larger size, the brighter nanodiamonds. It looks like you use 30 to 50 nanometers. What is limitation on size and why, if any limitation?
Yeah, so that's something that we're still investigating, and I think it's there are a lot of groups around the world looking into how nanoparticles in general are being packaged as they enter cells. So the way we get nanodiamonds into HeLa cells is just they just take them up. So we introduce them to the cell medium, and over time the cell will uptake these diamond nanocrystals. What we don't know exactly is, I mean, we have some preliminary data on this, but we want to look at it a little bit more, is how does the cell package it? And this seems to be very size dependent. And of course, you know, that depending on, you know, if, for example, if it gets packaged in a vesicle, the membrane might have very good thermal conductivity. So in that case, if you want to do thermometry, maybe you don't really care and you can afford to go for quite large nanodiamonds above 100 nanometers in diameter. Whereas if you're trying to measure, I don't know, say electric fields, you might be more concerned about actually having your sensor exposed, you know, to some other membrane, say the cell membrane or a mitochondrial membrane, and not want this kind of protective envelope around it. So I think there's still some work to be done on what are the size thresholds. And of course, it depends on shape and surface termination as well. So there's quite a lot of complexity there. And then the sort of target sensing modality of what, you know, do you care how it's been packaged? Yeah, so I think in some cases, absolutely, you know, going for larger nanodiamonds with more NVs is fine. And for others, it may be that we need to stick to smaller ones.
Do you have more questions? Maybe please.
Oh, can you hear me? Yeah. Hi, Helena. Yeah, really great talk. I had just a question on when you do these, say, temperature measurements, you showed these temperature jumps on, like, you have kind of maybe over minute control, but how long does your like say an actual measurement take? Because I assume maybe you do some signal averaging and things like that, if that makes. So how long does, say, an actual measurement take to do on say a single particle?
Yeah, so for thermometry in particular, our sensitivities are around like 1 Kelvin per square root hertz. So measuring for 1 second, we can get a precision of 1 Kelvin. So it sort of depends on what's the precision that you want to reach, will determine how long you have to measure for.
Mm-hmm. So I guess how long do you typically measure for? Is that?
Oh, I mean, our experiments can run up to hours in principle, if we want to track the changes as a function of time. So the limiting factor there is more the amount of green irradiation that we can afford to deliver to the cells, because of this kind of gelling up process that I mentioned earlier. So that's kind of, how long can, well, I mean, in principle, the diamonds don't bleach. So if you manage to not kill your worm or your cell, then, there's no limit. And in terms of the shortness, it relates to the sensitivity of what you're trying to measure. So if you want to measure a change, if there's a sudden jump you're expecting of like 10 degrees Celsius, then, you only need to measure for, I don't know, maybe 100 milliseconds, or tens of milliseconds to resolve that.
Okay. Oh, cool.
Does that make sense?
Yeah, that makes sense. I had maybe like one other small question, like for this, when you said there's 3-dimensional tracks, so you said it's confocal, so you're also tracking not just X, Y, but Z as well. Yeah.
Yeah, absolutely. Yeah.
So I assume you just avoid like artifacts from this by using like a very dilute amount of diamond, right? So like I'm always amazed by, like, you can track a single particle, and like if there was another particle that maybe interfered with this particle, how do you know you're tracking the same particle throughout the entire process is right? That's kind of the question.
Yeah, absolutely. I mean, you're completely right in that if there were two particles that came close to each other, the tracker would not know, which one's gone where, and it might hop from one particle to the next. So yeah, indeed, as you say, we use dilutions that are low enough so that we don't typically run into that problem. We're working on functionalization strategies where ideally they would be kind of more stuck to a particular organelle, in which case we could work with higher concentrations, and we would know that they don't move, and we can safely kind of stick to one. But you raised a good point on the Z side of things, which I think I just glossed over in the talk. So of course, with the orbital tracking, we can only do kind of X and Y tracking. And so the sort of Z component, because we do track in 3 dimensions, the Z component is done by using so 2 collection paths at different focal spots. So we're actually continuously collecting at 2 different heights in Z. And depending on, which one gets more counts, we know we need to either move up or down. So that's how the Z tracking works in our 3D tracking.
Okay. Yeah, thank you. Yeah, again, really great talk. It's really, really awesome stuff.
Thank you. Thanks, Nicholas.
Yeah.
So there is a chance to ask your questions. Don't be shy. Just Helena, obviously you think that kind of this nanodiamond technology, it would be great to bring it to the real world so that biologists can start to use it. Did you talk to biologists or do you interact with them? How complicated it would be for them, say, to start to use this single nanodiamond breaking inside cells as a method to do measurement of viscoelastic properties or temperature? Do you have any kind of feedback from biologists if they would be willing to start to use yours and from other groups where single tracking nanodiamonds in a focus?
Yeah, I think it's definitely moving into that direction where people are trying to simplify things down and simplify protocols. I have to say, our interactions have typically been such that we still bring the biological systems to our labs. We're in the physics of medicine building, so it's in the physics department, but there are a lot of people working on biological systems in the same building, so it's kind of an interdisciplinary space. But I would say there are, there are different levels of complexity here. So for example, purely optical thermometry was mentioned, and some of the reactive oxygen species detection that Romana Shirhago's group has pioneered, that can be done through purely optical means. So I think there are certain schemes that are easier to implement, and you could well imagine like a sort of packaged hardware that you just plug into your confocal microscope and off you go. And then I think there are more complex things that rely on perhaps pulse sequences and timing hardware that's a little bit more tailored. So I think as we go further and really identify some of these niche applications, I think there will be an opportunity space for developing sort of plug and play, easy to use bits of hardware. And I think for some of the applications, that's we're already there. And for others, it's we'll have to see what the opportunities are and then develop space.
And say within a framework of your work, if to use ensemble measurements, so definitely for some questions you cannot use it and you have to use individual particle tracking for external magnetic field orientation with NV axis. But do you see some applications where ensemble measurements which would simplify the picture are useful, say for temperature, temperature measurement for ensemble nanodiamonds within
So do you
light
mean
cells?
Do you mean ensemble NV nanodiamonds?
Ensemble particles, say
Ensemble
not individual particles,
okay.
but say you inject, say, some high concentration, they are consumed, and then you just measure average temperature of some dividing cells, for example.
No, absolutely. Yeah.
Temperature
There are-
rise and so on.
There are a number of groups that have implemented sort of wide field-based imaging techniques with cameras. So some of the Australian groups and Jerting Chu's group in Hong Kong, just to name a few. So that's absolutely possible to do sort of parallel acquisition. And also there have been demonstrations of sort of zero field ESR. So some of these sort of more complex schemes can indeed be realized in the absence of an external magnetic field. So I think it's all possible. There are technical challenges around that, but I think it's in terms of really generating a sort of a map of an entire cell or an organism. Of course, just looking at a single location or, you know, cycling through multiple locations, which is what we can do currently with the focal microscope. We can't in parallel look at several locations, but we can go from one to the next and then come back through again to look at over a longer period of time. But yeah, of course, using a camera would be a really good option as well. Yeah.
Do we have any more questions? Otherwise, so thank you, Helena, for your amazing talk.
Thanks for having me.
It was great to have you here. And thank you, everybody, for joining us today. Oh, Marka, is it just applause? Yeah, I think, yeah. Okay, great. Okay, bye-bye.
Thank you. Bye, everyone.